Hey friends, it's Mr. Rogers, and today we're going to just kind of make sure mitosis is still in our heads from 10th grade. For the most part, I'm not really going to test too heavily on this. This is one question that is, or one section that is on obviously your thing on Thursday and Friday this week. There might be a couple small questions about mitosis on the AP exam, but nothing too crazy. It's kind of something that just isn't covered in this class. So I just want to make sure that we feel so comfortable with it. So we go through the cell cycle. That is something that is covered, and we went through G1, S, and G2, and it's a healthy cell. Let's just assume it's a healthy cell. At the end of interphase, the cell kind of looks like this. And with our light microscopes, we really can't see a lot of the organelles inside the cell, but what we can see is what's going on in the nucleus. And we kind of see the nucleus here outlined here in the animal, here in the plant. And we can kind of see the DNA inside there. It's starting to form up, and that'll be the big part of what prophase is. And so there are technically 4 major stages of mitosis. There's 5 that is discussed on a regular basis in AP Bio and Advanced Bio. But 4 major ones, and those are prophase, metaphase, anaphase, and telophase. The 5th one is technically prometaphase. That is between prophase and metaphase, and kind of like how the name suggests, it's kind of like the middle ground between the two. There's a big step between prophase and metaphase. I mean, in prophase, the nucleus is kind of breaking down. The DNA is forming into chromosomes, and all of a sudden metaphase, the chromosomes are in the middle and everything's kind of gone from the nucleus. And that's a big jump. And so scientists have started naming the jump between metaphase to kind of make sure that people understand that, it takes time. It takes time, and there's some things that go on in between that are kind of, I won't say forgotten, but are just there. So for our class, we're just going to kind of focus, though, on the 4 and just kind of acknowledge that there is that 5th one in kind of between, but it's really just the transition time. So the first thing we got here is prophase. And prophase, we have mitosis officially beginning. This is the first stage that mitosis is actually happening. And what we have here is the centrioles. They begin to move to the opposite ends of the cell. We call them poles, the opposite poles of the cell, kind of like North Pole and South Pole. And these spindle fibers are starting to form. These are cytoskeletons components, and they're going to be used to pull the chromosomes apart, the duplicated chromosomes. So the chromosomes got duplicated in the S period of interphase, just so you remember. And so we have these chromosomes, and they're starting to form. They start to look more and more like chromosomes as we go along here. And these microtubules, the cytoskeleton, their job is going to eventually be able to move these chromosomes apart and split them. I will say what is interesting about these are the meiotic spindle, just in case if I forget, this is what a lot of cancer drugs and chemotherapy attack, is especially with a chemotherapy drug called Taxol. Taxol stops meiotic spindles from actually breaking down and pulling them apart. So imagine that the cell gets into metaphase in the next step, and then the meiotic spindles can't actually pull back and rip the chromosome apart. They're stuck. So the meiotic spindle is a huge part of how we attack cancer cells. And so during this prophase, centrosome is basically building these microtubules. That's kind of, I wouldn't say building them. They are the command center to build them. We actually can take centrosomes out, and the cell will still go through this process with ease. It's just a little more sloppy. So we like to see it as like a good leader. When you have a good leader, things go smoothly. When you don't have a great leader, the same things can still happen, but it might just not feel as smooth. An aster is a technical term for it's basically just a little piece that extends from each centrosome, like it says there. It's really going to be able to connect to the microtubules and kind of be the connection point, and where it's going to wind back up the microtubule and start the pulling process in later phases here. So this is kind of what they kind of look like. You're going to start seeing we got some spindle fibers forming, got some chromosomes forming. The nucleus is starting to break down. It's a big part of prophase. In metaphase, there's a big jump here, like I said. So this is where prometaphase would come in, where it'd be the last couple stages before it gets to this point. We have the chromatids, which are the technical terms for pairs of chromosomes. This is a really hard concept to kind of understand at times, but it's not extra chromosomes. They're copies of the chromosomes, and they're exact copies. And so it's the idea here is if I'm turning in a piece of paper or turning a multiple piece of paper into a teacher, and it's 4 pages long, my paper that I'm turning into like Miss Marks for English, but really it's just a copy of the first page. The next 3, I don't really have a 4-page paper. I have a 1-page paper with 4 copies. And so these chromosomes, they are copies and they're connected here in the middle. So this side is a copy of this side, even though in the picture they're not really copying-ish, but they are. Just let's assume they are. So in this example, in mitosis, whenever you see metaphase and say there's 4 X's here, truly a cell here, I know have 4 chromosomes because this is a copy of this guy. So when they finally pull, there'll be 4 chromosomes here, 4 chromosomes here. So they're meeting in the middle. In 10th grade, we always say M for metaphase for middle. You get the centrioles and spindle fibers connecting. You kind of see what they look like here. So these are the chromatids meeting in the middle. We get the spindle fibers. The spindle fibers are kind of hard to see here, but they're really easy to see in the animal cell right here. And you can see them meeting in the middle. It's chromatids. In anaphase, A for being pulled away. Chromatids are going to start pulling apart. So these microtubules start basically rewinding back up. I almost picture like a winch, like on a truck or some other kind of like tow truck, and you start winding the winch back up and it pulls the microtubules back in, and they're connected to these chromatids, so they're going to start separating them back out. And you can see that, again, like I said, this is a copy of this copy. So these 2 cells will end up being identical to each other, so it's not a big deal. They're being separated. So these 4 going this direction, these 4 going this direction. Towards the end of anaphase, you'll start seeing also something called a cleavage furrow start forming, where they'll start separating. Sometimes people call that, when that starts forming, the transition into telophase, which is the next one, or telophase, depending if you're British or English. So this is what anaphase kind of looks like. Plant cells here, kind of see it. Animal cells again, and you can kind of see it starting to go a little bit here. So this is like at the very edge of anaphase. And then telophase. So we're starting to get 2 new nuclei. So they're starting to form at the edges or the poles. We start breaking down the spindle fibers, and the centrosomes start kind of going back to normal. The chromosomes will become like thread again, and we're starting to get the end of mitosis. Now, again, this is called a cleavage furrow. This especially happens in animal cells. Okay, animal cells. I'll show you what happens in plant cells in a second. So it's separating out. You can kind of see they're forming. Plant cells don't really have the flexibility like animal cells do. They don't have animal cells have the cell membrane. That's it. We have cell walls here. And so what plant cells do is they're actually going to build a wall. So picture, some of you do probably have a sibling maybe in your room, but picture you share a room with a sibling, and you're fed up with it. And so what you're going to do is you're just going to put a wall up between the 2 and make it 2 different rooms. That's what's basically happening here. And from there, each cell will grow and the plant will expand. Okay. Now they can't grow that much because cell wall is kind of a limiting factor, and that's one of the reasons why plant growth usually happens at the very edges of plants, not in the middle. But it does occur in the middle, just very, very, very slowly and not much. Now, this process of actually separating into 2 is called cytokinesis. Cyto, cell, kinesis, to be moved. Okay, so this is really for the cell membrane to move inwards, especially for animal cells, and these 2 cells become their own identical cells. And so overall, these are the phases of mitosis. And I have this animals right here. I don't have prometaphase in here. Like I said, this is an older thing, but I mean, you could draw a picture of prometaphase easily into your 6, and that's what I asked you guys to do, I believe. So we have interphase here. You can kind of see the nucleus right here. Then we start separating out, making little things. You can't really see the spindle fibers in this one, but they are forming around here. You can see the spindle fibers here and the centrosomes connecting. Metaphase right in the middle. Anaphase being pulled away. Telophase, you're starting to get the separation of the two. And officially they're separated. Now they're both living an interphase life. And they're going to go through G1, S, and G2. Just to make sure we hit this home, G1 can be a long period of time. So don't think like, oh, they're just going to continuously keep growing. Some of your cells will be in G1 for months, years, maybe. A plant, this is what it kind of looks like. Interphase, same kind of look. Prophase are the chromatin starting to form. We have the spindle fibers here. Don't get confused by these. These are other plant cells. Spindle fibers. Okay. Metaphase, chromosomes in the middle, get pulled apart. Telophase, they're starting to build a wall. It's kind of harder to see in this one, but you can see the wall kind of forming. And officially, they make a wall in between, and they're an interphase. So that is mitosis. That's what I kind of need you guys to know. One of the main reasons why I need you to know is we do go over meiosis a lot. And meiosis is how you make sex cells. And the first part of mitosis, we'll go through this. Meiosis looks like mitosis with some variations, and meiosis happens twice. And so therefore, we're going to make one cell that is a diploid, which means it has the full set of chromosomes for a living thing, into 4 haploid cells. Mitosis takes 1 diploid and makes 2 diploid. What meiosis is going to do is take 1 diploid and end up making 4 haploid, or half of a cell of chromosomes, which are 4 sex cells. So that's where we're trying to go after break. And so we kind of do need to know mitosis, even though, like I said, it's not a huge part of this class. We kind of have to have an idea. So I hope that makes sense. I hope you're starting to get the idea. Besides that, you have your cancer project today. I hope you start that a little bit and work on that on Thursday, Friday. If you need any help, obviously let me know, and I'll get to you guys as quickly as I can, and I'll talk to you guys soon. Thanks, guys.